Excellent. So hi everyone, my name is Natalie Twine, and today I'm going to talk to you about genetic ancestry in the hunt for disease genes and the fight against COVID-19. So I'm here from CSIRO, Australia's national research agency, well known for the development of Wi-Fi and the Hendra virus vaccine. So genomics affects everything from how we look to our disease risk and indeed our behavior. So whether or not our lungs are healthy or carry cystic fibrosis based on the disease variant that an individual may carry. It also affects whether or not we dislike the taste of coriander. For example, 1 of 6 of us dislike coriander based on the genetic variant that we carry. And knowing your genetic ancestry is part of the puzzle to understanding and estimating disease risk. And because of this, we've developed a tool called Tribes, which is a population scale ancestry predictor. This is a pairwise relationship classifier, which is a cloud-based tool and substantially more accurate than gold standard tool King. And you can see the performance of Tribes compared to King on the right-hand side plot, where we have the true positive rate of Tribes in orange compared to King in blue, across a number of different degree of relationships, from 1st degree, which is a parent-offspring relationship, up to 7th degree, which is a great-great-great-great-grandparent. And by identifying relationships between individuals based on their genome, we can really increase our power to discover new disease variants. The application case we have for this is in ALS or motor neuron disease, where we've discovered new relationships as well as new disease genes. So this is work we published earlier this year in Nature's Genomic Medicine, where we've been able to connect 19 individuals from distinct families, different families, all suffering from ALS. And we've shown that they all have shared ancestry, so they have a common founder. So the clustering analysis on the left-hand side shows these individuals, where every individual is represented by a shape or node in the cluster, and their family of origin is shown by the color of the node. The edges between the nodes indicate that these individuals are identical by descent over the SOD1 gene, which is an ALS-causing gene. So here we've clustered these 19 distinct families, showing that they have a common ancestor and share this variant of the SOD1 gene. We also have, in the other 4 clusters, different variants of the same SOD1 gene, again showing that the individuals in the clusters share a common ancestor. Here it's important to know different variants of the same disease gene, because individuals can respond differently to the same drug depending on which variant of the disease gene that they indeed have. So we also have made Tribes scalable, which means that it can analyze very large data sets. So here we've analyzed 9,000 individuals, again, all with carrying the ALS disease. This is from the largest international consortium ProjectMinE data sets. So on the left-hand side, we can see the clustering analysis of individuals where every node or dot represents an individual. The line between the dots indicates the genetic distance or the relationship between the individual, where they could be 1st degree up to 8th degree. And again, we can see here individuals cluster based on their country of origin, where the country is the color. So for example, we have Dutch in pink, Belgian in black, and we can see them clustering closely. And then we have British in green, Irish in orange, and US in purple, also showing clustering closely together. On the bottom right, we have a Swedish cluster, which is very homogeneous and tightly clustered. And when we zoom into this cluster on the right-hand side, you can see we've been able to identify 46 novel relationships up to 5th degree between a cluster of 22 ALS patients, all originating from Sweden. And by using the shared regions between these individuals, we can really narrow down the search space for looking for disease variants that drive the disease in these individuals. So moving from humans to COVID-19, we have also used genetic ancestry in the fight against the COVID-19 pandemic. So again, work we've published earlier this year, we've taken the genetic fingerprint of 181 SARS-CoV-2 virus strains. And here we've looked at the genetic distance between them in 3D space, which you can see on the right-hand side. And this information was used to pick the most relevant viral strain for vaccine development in Australia. So we've also developed a website which hosts all this information and analysis, and it's updated daily from GizAid based on any new viral strains that are uploaded over the previous 24 hours. So I encourage you guys to go and check it out. So I'd like to thank the rest of the team in Sydney here. And also here's a QR code for anyone who's interested in checking out other technology. I encourage you to go and look at our website. So if there's any questions, please feel free to ask.
Thank you. Thank you. Very interesting talk. So are there any questions from audience? Okay. Yeah, so I think that I'm very impressed in your talk. So, but I'm not sure the so eventually can use the real ancestor of the source of the COVID-19 strain. So how so can you succeed the origin of COVID-19 strain? So do you already identify some original strain of the COVID-19?
I guess, so what we wanted to do was pick the strain that's most relevant or more prevalent in Australia, because we were developing, we don't develop the vaccine ourselves, but we have within CSIRO groups that work on vaccine development. And we wanted to find a strain or a few of the strains which were sort of most representative of the majority of the strains that we are seeing in Australia. And so we did this sort of genetic analysis to compare the strains to each other. And then we've expanded it out to look at all the global data, because we built the technology to be able to do this. And it uses Amazon Web Services to pull in the data from GIZ8 every day, so that anyone can go in and check out, you know, the viral strains that's relevant to their country of origin.
Okay, and does your analysis, can you analysis the relate to the some so severe phenotype like toxic or some more disease related, or I'm not familiar with that COVID-19, but can you relate your analysis to the phenotype of the COVID-19?
So from what I understand, in the GIZ8 data, the GIZ8 data that's publicly available, and that's what we built this analysis from, we don't currently have any subphenotypes other than whether or not the individual was affected. But I do know in the UK Biobank data, they are having different phenotypic attributes for individuals captured in there. So that's, I know there's a lot of work going with the UK Biobank data to say subphenotype, to look at individuals who either have a sort of protective effect against the virus. So individuals are in the high risk groups, so over 70, who haven't got very sick from the virus, or indeed individuals who are low risk groups, they are young, but have got very sick from the virus. So yes, not in the data set that we've analyzed, but if you go and look at the UK Biobank data set, it is possible to get the sort of subphenotyping and look at risk variants that drive a specific phenotypic outcome.
Okay, okay, thank you very much. Okay.
No problem.
Yeah, thank you. So if not, so let's move on to the next talk. So thank you very much.